continue our session with next speaker, Dr. Michael Alexanian. Dr. Alexanian is an assistant investigator at Gladstone Institute and an assistant professor in pediatrics at UCSF. He received his bachelor and master degree from the University of Florence in Italy and a graduate degree in stem cell biology at the University of Lausanne in Switzerland. During his doctoral study, he investigated the role of non-coding RNAs and long non-coding RNAs in potency and early cell phase specification. During his postdoc at Gladstone, Dr. Alexanian discovered gene programs relative to cellular plasticity and in the context of heart failure. Please welcome Dr. Alexanian.
Thank you. Thanks a lot, and thanks for the introduction. I'm happy to see this wonderful community all together. So I started my lab around a year ago here at Gladstone, and what is the general questions we're interested in the lab is really trying to understand what are the molecular mechanisms that govern cell states in normal and abnormal physiology. And if you think about the heart, no matter which pathological insults affects the heart, whether it's MI or untreated hypertension or genetic mutations, these stress signals, they all converge at the nuclear level where specific enhancers are activated to support the transcription of genes and these pathological gene programs that drive changes in cell states. And all these processes ultimately can lead to organ dysfunction. So my lab really studies how chromatin receives, processes, and amplifies these stress signals in cardiovascular disease, but specifically in heart failure. Of course, this audience doesn't need any introduction on this disease, but just to remind you that it's a very common clinical condition associated with high morbidity and mortality. The therapies that we have available are not great, and this really highlights the imperative need of identifying new therapeutic avenues. Now, we heard a lot about cardiomyocyte biology, but there's many features of heart failure, and two of them are definitely inflammation and fibrosis. Now, fibrosis is thought to be the result of the uncontrolled and pathological activation of fibroblasts that transition into an activated fibroblast state or myofibrillous state. And this here you can see an image of a human feeling heart with this fibrotic response all over. And inflammation and fibrosis have been thought to be highly intertwined for quite a long time, but the molecular mechanisms that govern how these two tissue processes are talking to each other are not really well understood. Now, as someone interested in gene regulation, heart failure, as we have heard already, it's really associated with the activation of specific pathological gene programs that drive maladaptive remodeling, and within these programs, we are very interested in the role of a family of epigenetic reader called bromo and extra-terminal domain proteins, BET proteins. That's this, we think, a really critical regulator for activating these genes that drive disease pathogenesis. Now, BET proteins are thought to be activator of transcriptional and chromatin states, and you might have heard of BRD4, which is definitely the most studied and understood. What these BET proteins do is that in the context of cardiac stress, they bind and activate specific enhancers, which in turn activate the transcription of these stress-activated gene programs, these pathological gene programs that drive tissue remodeling in the heart. And we have exquisite modalities to block BET-dependent transcription in vivo with small molecule compounds like JQ1. JQ1 will displace BET proteins away from their chromatin targets, and as a consequence, this will dampen the transcription of these stress-activated genes. Now, we've known for now around a decade that the inhibition of BET proteins improves cardiac function across several murine models of heart failure. This is work initially in the lab of Saphalder and Tim McKinsey. This is an experiment that we ran a few years ago where we trigger heart failure with the transverse aortic constriction. So this is a model of pressure overload heart failure. The LV ejection fraction is in the y-axis, x-axis is time. And you can see here in red the TAC animal, this classical drop in function. The sham is the control in green. And here in blue is what happens with a BET inhibitor. We start injecting the mice daily via IP, 18 days post-TAC, where the cardiac dysfunction is already established. And you can see this pretty striking recovery, right? The function basically goes back to sham. Now, this is not just potent, but extremely reversible, as when we stop treating the mice with GQ1, the function goes back down, really highlighting how we can basically toggle the function up and down by starting and stopping the BET inhibitor. And this has been fascinating and intriguing to us for quite some time. Like, how does it work? What are the cell types? What are the gene programs and molecular mechanisms that are underlying this process? And to start addressing this, we launched this large single-cell omics analysis in which we perform single-cell RNA-seq and ATAC-seq to look at transcription and chromatin accessibility. Now, one of the ideas that we had was related to the function of BET proteins that are so important regulators of chromatin states. So that's the approach that we decided to take. Why don't we use single-cell ATAC to define a catalog of putative regulatory elements in each of the major cell types, and then correlate the accessibility at this enhancer. Now, accessibility is a proxy for activation with ejection fraction to identify, like this schematic figure shows, enhancer with accessibility. It's, for example, anti-correlated with function, which will mean the function goes down with TAC. These enhancers, they open. We give the BET inhibitor, the function goes up, these enhancer close. We withdraw GQ1, the function goes back down, and these enhancer open up again. And the positive trend, it's the opposite. Now, to make a very long story short, we found that the cardiomyocytes didn't have a lot of these regions, were not very sensitive to GQ1, at least at the chromatin and transcriptional level. But the cells that really stood out as being very sensitive were the fibroblasts. And we found that one of the most anti-correlated enhancer of all the fibroblast genome was proximal to a transcription factor called MIOX1. This was already pretty exciting. There is a TF next to one of the most regulated region. This TF, it's called MIOX1, very important in early development, but really poorly understood in adult disease, specifically in heart disease. And we went on doing a series of in vitro experiments in which we used TGF-beta to trigger a profibrotic response and define the specific enhancer that control MIOX1 stress induction, and then went on describing MIOX1 as a transcription factor that binds and promotes profibrotic function. So we believe that MIOX1 could serve as a switch to activate the fibroblasts from a quiescent to an activated state. And if you're interested in what my lab is now doing to understand MIOX1 function in vivo, visit Ada's poster this afternoon in the poster session. But now I want to tell you in the remaining time about an unpublished story that really tries to address one question that it's important and to me very fascinating and indeed very relevant to the story I just told you, which is how are the fibroblasts activated in the first place? What are the stress sensing mechanisms that initiate the transcriptional activation of the fibroblasts? Now, this is very relevant in heart failure, but it's relevant in many other diseases where fibroblasts are activated. Lung fibrosis, liver, kidney fibrosis, and of course now a lot of traction in understanding cancer-associated fibroblasts and their interaction with, for example, immune cells in tumor microenvironment. So what is the signal that activates the fibroblasts? Now, something that stood out in our previous data set that I just described you with TAC plus and minus GQ1 was that beside the fibroblast, another cell population that was very sensitive to this perturbation were the myeloid cells. And here I'm showing you a volcano plot which depicts the analysis of correlation between ejection fraction and this time not ATAC-seq, but gene expression. So basically, we're measuring by single-cell RNA-seq in the myeloid population which gene has expression that it's most anti-correlated or positively correlated with ejection fraction, which will mean again, when the gene goes up, the function goes down. When the gene goes down, the function goes up. And I want to draw your attention on the left part. These are the negatively correlated gene. And one of the most anti-correlated gene in the myeloid population was CX3CR1 that caught our attention. Why? CX3CR1 marks both resident cardiac macrophages, so the ones that are embryonically derived, as well as the monocytes that infiltrate the heart upon injury and tissue. So we were intrigued about this dynamic regulation, and we thought, what would happen if we had to inhibit bed-dependent transcription, specifically in the CX3CR1 population, which of course we cannot do with JQ1, right? JQ1, it's not cell type specific. So we need the help of mouse genetics. Now, if you want to know more about the details of the story that I'll summarize in the next 10 minutes, we have a preprint on this, and Kirsten in my lab has a poster this afternoon with more details. So to address this question, we took a CX3CR1 inducible CRE and crossed it with the BRD4 conditional allele that we generated a few years ago. I remind you, BRD4, it's one of the bad proteins. We performed sham and tuck surgeries, and first of all, we looked at ejection fraction. So again, systolic function is here in the y-axis. I'll draw your attention to this day 30 post-doc. The blue group, it's where BRD4 has been deleted in the CX3CR1 population, and you can see significantly higher ejection fraction as compared to both the CRE control in gray and their leader-mate flux control in red. So deleting BRD4 specifically in this immune population is protective in this model of heart failure. How does it work? Again, we thought to use single-cell omics to understand what were the underlying consequences of this genetic perturbation. We perform single nuclei RNA-seq and anatoxic, and first look at the RNA-seq expression. This is the UMA plot. The color code is control is in red, and the knockout of BRD4 in the CX3CR1 population is in blue. You can see all the major cell populations. A few years ago when we did this was 2 years ago, our ability to get the nuclei from the adult myocytes was not great, was suboptimal. So that's probably explained why we have less myocytes. Now we're much better than that. But what was important is when we run differential expression analysis between the control and the knockout, and then rank the different cell compartments for the number of dysregulated genes that they were having, we found, not surprisingly, the myeloid had the highest number of dysregulated genes. This made a lot of sense, right? CX3CR1 is expressed only in the myeloid population, so this is the genetically perturbed cells. But what was happening in the fibroblasts was very, very intriguing, right? They do not express CX3CR1, though they had changes in gene expression that were similar to the one in the myeloid cells. And we focused more on the changes in the fibroblast transcriptome. So these are the fibroblasts reclustered. Again, same color code. Red is control, blue is knockout. And here I'm showing you the expression of two profibrotic genes, periostin and MIOX1. Now, MIOX1, I already told you, it's this gene associated to fibroblast activation that we identified a few years ago. Periostin is really the classical myofibroblast marker in heart failure. And you can see how the expression of these two profibrotic genes and many other hundreds of profibrotic genes was strongly downregulated when BRD4 was knocked out in the immune population, together with fibrosis that we measure with histological analysis. So let's take a step back now. CX3CR1, it's not expressed in the fibroblasts, does BRD4, it's not gone from that population, but there are striking changes in the fibroblast. So this reflects a non-cell autonomous mechanism that was under the control of BRD4 in the CX3CR1 population. How would we go around finding which signal that was? Well, of course, we were looking for genes that were expressed in the CX3CR1 population that encoded a secreted factor, as we were interested in paracrine signaling, and also to genes that were upregulated in disease in heart failure and downregulated with both approaches, the BET inhibitor and the knockout of BRD4. And when we ran this series of analysis, it became pretty evident that a promising candidate was interleukin-1 beta, I1 beta, that encodes for an important pro-inflammatory cytokine. And here you can see that IL-1 beta, exactly like CX3CL1 was one of the most anti-correlated gene in expression with ejection fraction, looking at the myeloid cell population. So we had two questions. The first one was, how does BRD4 control the activation of IL-1 beta in the immune cells? And the second, how does IL-1 beta affects fibroblast states? So to address the first question, we looked at ATAC-seq in the IL-1 beta locus. Here you can see the gene. Below there is single cell attack in the monocyte macrophages in sham and TAC. And here you can see there are many peaks that are accessible. And as we and other people have noticed, single cell attack great to identify putative regulatory elements, but sometimes you don't really see a dramatic changes in accessibility, although this gene in transcription, we know it's dramatically modulated. And moreover, accessibility doesn't really mean functionality, right? The fact that something is accessible doesn't mean that it's doing anything. So to really understand how this locus was more specifically regulated in stress, and knowing that BRD4 was likely very important, we made a new mouse model in which we knocked in a 3x flag in the N-terminal of BRD4. We took these mice, performed sham and TAC, sort the CX3CL1 population, and perform anti-flag cut and run that allow us to map genome-wide BRD4 occupancy in a number of cells that it's pretty small. It's 50,000 cells, what I'm showing you right now, which, as you know, would be impossible with a classical ChIP-seq approach. And now, when we were doing this, it became pretty evident that in this very large enhancer, there were indeed two sites that were more bound upon TAC by BRD4, and I'm highlighting them here, PIC5 and PIC6. Now, this was still not telling us anything about whether these regions were important. So we took CRISPR, we used CRISPR in row macrophages and delete each one of these peak in the IL-1 beta locus independently, and then look at IL-1 beta expression upon LPS, which notably activate a pro-inflammatory response and IL-1 beta activation. So IL-1 beta expression is here in the y-axis, x-axis are all the clones. You can see no LPS, no IL-1 beta expression. This is known. The first two columns here in red are the wild type with LPS. These are clones with a guide RNA control. And you can see that most of the genetic deletion, they don't do anything. IL-1 beta still goes up, but when we delete either PIC5 or PIC6, it's where the cells almost lost all the ability to upregulate IL-1 beta upon LPS, suggesting that these two BRD4 bound enhancers are really the essential regulatory elements for controlling IL-1 beta and stress. Now, the next question was, how does IL-1 beta affect fibroblast states? So we ran a series of behavioral assays on human cardiac fibroblasts to prove that IL-1 beta synergized with TGF-beta, which is the classical profibrotic signaling molecule, to activate even more profibrotic response. Again, if you want to know about some details, go at Kirsten Poster later. But how does it work? What are the downstream consequences of IL-1 beta in the fibroblast? What's the molecular mechanism? We ran a series of RNA-seq where we asked the following question. Can we identify genes that are upregulated by TGF-beta, but there will also be more expressed when IL-1 beta is added over TGF-beta as compared to treating the cells only with TGF-beta? And we found a series of hits that were very interesting that had this feature. And again, this gene MEOX1 came up again with this unbiased screen, and we were very intrigued, right? We knew that MEOX1 was very sensitive to TGF-beta. That was what we studied in our previous story, but how does this pro-inflammatory signal affect MEOX1 expression? So we looked at MEOX1 locus. This is the gene here. This is this downstream enhancer. This is H3K27 acetyl in human cardiac fibroblast. The enhancer that I'm highlighting here, that's the synthetic region of the mouse enhancer that we prove being the essential regulatory element to drive stress-induced activation of MIOX-1 in mouse heart failure. This enhancer was enriched with motifs for P65, which is the transcriptional effector of the NF-kappa B signaling, which is notably activated by IL-1 beta. We performed ChIP-seq for P65 and found that indeed this transcription factor was binding this enhancer. And then we wonder what would happen if we had to deplete P65 expression. We've done this with an siRNA for this factor, and we measure MIOX-1 expression. You can see the kinetics with an siRNA control. It goes even higher with both molecules, like the RNA-seq had demonstrated. But more importantly, when we deplete P65, the cells strongly downregulated MIOX-1. So it seems that it's really a factor important for driving MIOX-1 expression, even only with TGF-beta, which we don't fully understand. Now, we went back, this is the last dotted slide, to the in vivo model of pressure overload by attack and wonder what would happen if we had to inhibit IL-1 beta in vivo, which we have done, treating the mice with an anti-IL-1 beta antibody. So we looked at ejection fraction. At the beginning, again, I'll draw your attention to the day 30 post-hoc. The red, it's the IgG-treated animal, so this is a control antibody. The blue are the animals that got the neutralizing antibody for IL-1 beta. You can see a significantly higher ejection fraction, but more importantly, we were interested in understanding how the fibroblast states were modulated. In this context, we performed single-cell RNA-seq. I'll show you again just the two genes that are sensitive to anti-IL-1 beta treatment. Again, these two genes, periostin and MIOX-1, are example of profibrotic genes, but there are hundreds of these genes that behave in the same way. Indeed, the anti-IL-1 beta treatment decreased the expression of periostin and MIOX-1 and also decreased fibrosis measured by histological analysis. So what I told you is how we attempted to really integrate modulation of cell states with either a small molecule approach or a cell state-specific BRD4 inhibition in a specific immune population, and then integrate this with single-cell omics. This can teach us not only what's happening in the population that we're genetically perturbing, in this case the CX3CL1 cells, but even more importantly in this context, what's happening in the other cell population that are not genetically perturbed. And I told you about how BRD4 controls the expression of IL-1 beta through specific enhancer, and how IL-1 beta is able to promote profibrotic function. Of course, the mechanism will be very complex. We believe that MIOX-1 is one target. This enhancer in the MIOX-1, it's something that for us it's very intriguing because it's an example of a regulatory element that can process, like a regulatory hub, multiple signaling pathways, not only classical profibrotic stimuli like TGF-beta, but also classical pro-inflammatory stimuli like IL-1 beta, and this provides a mechanistic explanation on how inflammatory cues can modify fibroblast states. Of course, the biology is much more complex. There will be a lot of other enhancers in the fibroblast genome that are affected by this signal, and even within the same enhancer in the MIOX-1 locus, there will be signal coming from other cells like myocyte or vascular cells that are able to modify the expression of these genes. And this is really one very active research program that we have in the lab, using an integration of single-cell experiments with in vivo physiology to really go after this important question: what are the mechanisms that govern pathological cellular crosstalk in heart failure, and can we find ways to inhibit this crosstalk for therapeutic application? I'll just finish here thanking the people in the lab. They're all here, so I hope you can meet them today. There are a lot of people that were important and are still important at Gladstone, but I want to specifically highlight Deepak and Arun, who's been leading these efforts with me, a lot of collaborators, the funding, and of course, I'm happy to take any questions. Thank you.
Thanks, Dr. Alexanian.
Hi, fantastic story. So you have this previous story that focuses more on fibroblasts, and then you have this more recent one that is more immune fibroblast crosstalk centric. And so I was just wondering if you've done like a head-to-head comparison between just the JQ1 treated TAC hearts versus the ILB1 or ILB anti-ILB treated hearts, to see which one kind of sort of dominates.
Right. It's a good question. We haven't formally attacked that question, meaning take the data set and compare them. The changes in ejection fraction are a bit different, right? There's basically nothing we do that does what the BET inhibitor does, right? The BET inhibitor basically brings back the function almost to sham level. The IL-1 beta inhibition doesn't do that. So it's a strong effect, but it doesn't do that. So in terms of changes in gene expression, indeed, we do find that the decreased pro-inflammatory signature that we find in the myeloid cells, it's pretty comparable when we look at the BET inhibitor and the IL-1 beta application. So at least in the immune cells, we believe that it's pretty comparable. Now, I want to also make the point that BRD4 will control a lot of other cytokines and chemokines that are important for pro-inflammatory signature that are not just IL-1 beta. But that's a very good question, and I think we should just attack this more systematically with a computational analysis.
Hey, great story. Thank you. I have a question about that IL-1 beta enhancers that you described. And I'm sort of intriguing that deletion of either peak 5 or peak 6 resulted in a complete loss of gene expression. And I'm wondering how you're imagining that. And it is sort of also notable that peak 5 is right where one might think a proximal promoter would be. So do you think you knocked out the promoter and the enhancer, and you're calling them both enhancers? If they're both enhancers, how do you think that's working?
No, that's a great question. So let me first clarify one thing. We were very concerned about the promoter being nexus, so we've done a lot of control to ensure that the promoter, it's not affected. Now, with this scale, given that LPS is 1,000-fold, you don't really see this difference, but actually peak 5 and peak 6 don't completely abolish it, but brings it back to around 20 to 50-fold. We have done the experiment in which we deleted one and then the other one in the same cell type. So now they're both gone, and now it goes completely to zero, like zero, okay? So there's still some response when either one is present, but when both are gone, there's absolutely no response.
Yeah, but even still, even if there's a scale issue, they're not functioning additively. They look like they're functioning multiplicatively in some way, which might suggest that those two enhancers are talking to each other in addition to talking to the promoter. And I wonder if you...
Probably. We haven't, yes. So if you're referring to, doing some chromatin loop analysis, we haven't done that yet, but it is possible that they're talking to each other. Also, they're pretty close. So an assay that would discern that it could be challenging, but yes, there could be definitely ways these two regulatory elements are talking to each other.
Thanks.
Great talk. A question about the relevance to human heart failure. Have you looked at any multi-omic data sets that any of these processes are actually applicable, or you see the same effect in human heart failure?
Yeah, it's a great question, very relevant. So this story I just presented, it's in review, and it's in review together with another story that, interestingly, basically it's looking at very similar mechanism in human heart failure by Corey Levin. So they do see there's a specific population of monocyte-derived macrophages that are affecting, that are expressing IL-1 beta. With computational analysis, they predict that the receiving cells are the fibroblasts, and have done other computational analysis to map that the kind of transition that we are mechanistically dissecting in this story, they seem to be very relevant in the context of human heart failure. Now, regarding the activation of MIOX-1, looking at those at that data set, it's pretty striking that MIOX-1 in their data set, it's the most sensitive transcription factor in the population of profibrotic fibroblasts in human heart failure that they computationally imagine and predict are the targets of IL-1 beta. So I think that it could be relevant. Of course, we would have to do more experiments. And, as you know, there is a lot of attention on I1 beta therapy is not for heart failure, but for atherosclerosis. But there are some evidence that inhibiting I1 beta would have an effect in heart failure.
Thank you.
Mikey, over here.
Yeah.
Great talk. I have a couple of questions. So when you gave the IL-1 beta antibodies in vivo, did that reduce the macrophage recruitment to the heart? And could it just be that there's less macrophages maybe in that context?
Yeah.
And I'm just also curious, maybe I missed it, but was the MEOX1 actually linked to a functional effect in the fibroblast or in the fibrosis?
Yeah, so to answer the first question, we haven't done that, right? We haven't done flow to address whether anti-IL-1 beta would change the infiltration. My guess would be yes. Okay. The second question is related to MEOX1. So we've done some in vitro assays that are indeed in the paper in which we take human cardiac fibroblasts, treat them with TGF-beta and I1 beta, measure collagen contractility, and of course it goes up with both molecules. And when we knock down MEOX1, it's this contractility. It's basically abolished again. This will be in the poster this afternoon. For in vivo, we made this flux animal right now because it wasn't existing, and this is something that my lab is very interesting. Is it MEOX1 a nodal regulator of fibrosis in vivo? I guess that's an imperative question that my lab has to address.
Thanks.
This is a beautiful story, and it's just so great to see. I was particularly intrigued by your comparison of looking at the LVEF and then correlating that to the single-cell data. One thing I'm curious, though, is if you have acute pressure loading to an LV, you're going to drop LVEF, even though the contractility, when you measure on pressure volume loops, is the same. And so if you're normalizing the LVEF, you have to assume that the contractility is actually even hypercontractile in that LV. So I'm just curious, is he actually reflecting, in the JQ1 story, are you actually showing hypercontractility? Have you done pressure volume loops and looking at that? And then also just in sham mice getting treated with JQ1, do you see increases in LVEF?
Yeah. So we don't see it in the sham. I think that this was addressed in some of the first publication by Sepp Halder that showed no changes. So we were not particularly concerned about that, but it's true that it could affect our system and our way we're thinking about also the dynamic response in the chromatin dynamics that I've pointed out. So
if you relate that to the large clinical trials that then subsequently show no benefit in human heart failure, what's your thought? How do you put this all together when now you're kind of looking at this stage?
For the anti-IL-1 beta?
Yeah, or IL-1 beta, then also the BET inhibitors.
Yeah, so BET inhibitors, there wasn't a large clinical trial for heart failure yet. I1 beta, there was a large clinical trial for atherosclerosis, which, as you guys know, was dropped for reasons that were also related to the inflammatory reaction and sepsis. There was a subgroup studies that did show effects in patients that had human heart failure. So I think that this line of investigation and maybe the other paper that looks at human heart failure might restart some more interest in targeting I1 beta human heart failure. But of course, we know that I1 beta is just one factor, and there will be many more other players that can affect heart function. So there's a lot to know. But for the BET inhibitors, I'm not aware of any large clinical studies in heart failure.
Great, thanks.
Thank you.